University of Chicago. Today I feel really privileged to share a bit of a legacy piece out of the Westgate Lab. And before I get started, I want to draw attention and acknowledge my collaborators on this project that have contributed significantly to what I'm going to be talking about today. So without further ado, I'd like to introduce the Chaetodontid and Pomacanthid. Angelfishes and butterflyfishes are two of the most conspicuous, ecologically important, and well-studied families on tropical coral reefs. So at first glance, there's a lot of similarities between these two families. They're brightly colored, often patterned. They have this terminal jaw, also this lateral compression of the body. However, if you dive into this rich literature on these groups, there's a lot of really notable differences with respect to life history, ecology, as well as anatomy. So today I'm going to be building off of the excellent literature that exists for these groups and talking about two main projects that we've been having going on. The first is about phylogenetic relationships, both among and between Chaetodontid and Pomacanthid. And then the second part of my talk is going to be a discussion of the evolution of cranial functional morphology in these groups. So again, as I mentioned before, there's a very rich literature about these two families. We're certainly not the first to investigate the phylogenetic relationships of these groups. I want to draw particular attention to really fantastic work that's come out of David Bellwood's lab, Leo Smith's lab, as well as previous students in the Westgate lab. Today, I'm going to be presenting the most densely sampled phylogeny for these families together that has been created to date. So we added taxa, we added gene sequences to come up with a total of 194 species with pretty good representation from the two families, as well as squamopene outgroup taxa. Our supermatrix was from Sanger sequencing that several folks did, and we also mined a lot of stuff from GenBank, concatenated everything into a supermatrix that serves as the input for all the phylogenetic analyses that follow. And this is kind of our pipeline that we used. Today I'm just going to be exploring the Bayesian analyses that we came up with. So without further ado, this is our Bayesian consensus tree. And just to orient you, up here in blue, we have the Keats, or the butterflyfishes, and at the bottom in red, we have the angelfishes. We reveal very strong support for the monophyly of these two families, and we also reveal very strong support for a sister-family relationship between these two families. This is significant because other folks in somewhat recent mitochondrial analyses of phylogenetics have found that scatophagy within the squamopene lineage sometimes pops up in between these two families. So now to kind of break this down into a way that's potentially a little bit easier to look at, first I'm going to talk about the angelfishes, so Pomacanthid. Again, we find very strong support for the monophyly of this family, strong support along the backbone of this tree with some resolution problems, both in terms of topology and supports kind of at the tips. However, if we break this down in terms of kind of genus by genus within the family, there's a lot of support for the currently accepted taxonomic designations. So for example, we have good support for Chaetodontid, Pomacanthus, Polycanthus, as well as Genicanthus being natural monophyletic genera. However, also notable that Apomixes and Centropyge, and kind of this big radiation right here, still some problematic taxa. Moving over to the butterflyfishes, much the same story. We get good support for the monophyly of the group, strong support along the backbone, some problematic issues kind of up near the tip. Again, breaking this down, we find very good support for the bannerfishes at the base of the tree. However, actually within the true butterflyfishes, there's a lot of problems here. We do recover some good support for the traditional subgenera within the Chaetodon radiation. However, I think when we go to publish this, we're going to strongly support that we adopt a new taxonomic classification that is identified by these 8 different clades that are very well supported by our data. Of course, right now they're not named anything too exciting, but when we look at color pattern, diet, and biogeography, perhaps we'll come up with some more descriptive names. So just to kind of sum this up, our robust phylogeny supports the monophyly of Pomacanthid and Chaetodontid. We also find a very strong support for the sister-family relationship of these two groups. However, I think it's certainly worth looking into the scatophagy, putting some more of these squamopene outgroups within an investigation of the phylogenetics of these groups before we make some taxonomic revisions. So now moving on to the second part of my talk, I'm going to talk about the business end of these fishes. I'm really interested in feeding functional morphology and behavior. And again, there's a lot of work that's been done on this for these particular fishes. In particular, if you're interested, I highly recommend you check this stuff out. But the main features of, again, kind of the business end, the cranial features of pomacampity and Chaetodonty have to do with the brush-like teeth that are used for scraping algae. They have a unique configuration of the adductor mandibulae muscles, which function in closing the jaws. And they also, well, not all of them, but many of them have an intramandibular joint. This is an extra jaw joint that these animals have on their bottom jaws. So, when it's present, it occurs in between the dentary and articular, and you can think of fish lower jaws as being a rigid beam around which it rotates around the quadrate articular joint. So it kind of looks like this hair clip right here. Whereas if you have an intramandibular joint, you have this extra joint right here, which during jaw closing, you have an extra region for flexion, which is particularly advantageous when the jaws are fully protruded during feeding behavior. Obviously, some very direct functional consequences, perhaps because of this configuration. The thing that's lacking from this very rich functional morphology data set is a comprehensive quantitative comparative analysis within a phylogenetic context. And so this is a growing data set at this point in time. This is kind of what we're working with right now. We're able to get into the very wonderful collections at the Field Museum of Natural History in Chicago, do a bunch of dissections, take pictures of all of our dissections. And then we came up with which you can't really see, but we came up with a novel landmark protocol, which outlines the shape of the cranial features as well as the functionally relevant locations. So for example, the outline and trajectory of jaw closing muscles, the location of jaw joints, etc. Then, with the total configuration of our landmark protocol, we subjected it to a Procrustes fit and then did a slew of very commonly used geometric morphometric analyses interested in the evolutionary relationship of form and function. Really great way of estimating bite performance and jaw biomechanics are by creating a jaw model. So essentially, if you connect the dots in our landmark protocol, we can come up with these very simplified measurements that give us a way to approximate various measures of biting performance in the group. And then because we have a pretty robust phylogeny for the group, this is essentially just a pared down version of what I presented earlier, we can use this to analyze form and function in this group within a phylogenetic context. So this is our phylomorphospace plot. A lot going on here. Before we get going, I should mention we did run a lot of tests to see if size and phylogeny are significantly influencing shape pattern evolution. And we found that both of those things are actually very significantly contributing to the structure of our data. So everything that I'm going to be showing you from here on out is within a phylogenetic context and also uses the residuals of a regression that we did of Procrustes scores against log centroid size. So a lot going on here. This is a beautifully messy plot. But first and foremost, from the roots of our tree here, there's a burst of morphological variation. And we can see here that very strongly and significantly along PC axis 2, angelfishes and butterflyfishes are occupying unoverlapping regions in phylomorphospace. What else was I going to say here? Also, I think kind of the messy parts of this graph are very interesting. This is kind of indicative of potentially some structural divergence or convergence going on. Again, as we expand this data set, especially in the angelfishes, we can look into those potential dynamics a little bit more closely. So what do these PC axes actually describe in terms of shape features? We can use thin plate spline to analyze the shape features that scale to these two PC axes, and this is just kind of a rundown quickly of those. So along PC axis 1, the lowest score, the highest score fish. The most obvious thing is an elongation and kind of a skinny-faced organism on the lowest PC scores, whereas on the far right-hand side, the highest PC scores, kind of you get a short, blunt face. On PC axis 2, a lot has to do with kind of the occipital crest, the dorsal regions of the cranium significantly being rearranged. So what about diet? Diet is also something that in the functional morphology literature has been shown in many instances to be directly affecting shape variation. Again, this is a work in progress, but if we code our data for diet, there is some signal here, some interesting things going on. So for example, we can see over here here in purple are the soft coral-eating fishes. They're occupying a very thin sliver of phylomorphous space. Same thing goes for the obligate corallivores that are eating hard corals, whereas fishes that are eating invertebrates occupy this massive region of phylomorphous space. So this is indicative that there's more ways to be put together and be functionally capable of eating invertebrates, whereas if you are eating coral, you've got to be designed a certain way to overcome the constraints of eating that specific thing. So getting back to, okay, we know that there's a lot of shape diversity within these two families. What does it mean for the actual biology of the animal? Today I'm just going to be presenting kind of a first character for an approximation of the jaw biomechanics of these groups. We're using jaw mechanical advantage, which is essentially based on the location of jaw joints, tip of the jaw, as well as muscle insertions. You can calculate this ratio of in-lever to out-lever and get an approximation of how well force is amplified through a jaw system. I should mention that this is a simple calculation. I mentioned before the presence of the IMJ joint, that flexion joint in the lower jaw. What I'm going to be presenting today doesn't yet take that into consideration. Moving forward, we absolutely will, but just so you know, this is essentially a calculation that assumes that the IMJ is kind of locked into place. So take it with a grain of salt. This is what we get. So there's a lot of diversity going on here. So on your left are the angelfishes, and then here are the butterflyfishes. So again, a lot of variability both within and between these two families. Most notably, within the angelfishes, we get this evolution of pretty high upper jaw mechanical advantage, which is kind of interesting for fishes. And again, going back to the phylogeny, what we can do is actually map these characters onto the tree. And again, we get something kind of interesting. There's this reoccurring theme in especially the fish functional morphology literature, kind of local divergence with global convergence. And so if we just look, for example, at this evolution of high upper jaw mechanical advantage, this has evolved independently at least 6 times within this lineage. So I'm really interested in kind of fleshing out this story from both an evolutionary as well as a functional perspective, adding more characters and seeing what we get. So to sum up here, size, phylogeny, as well as diet have contributed to the shape variation and functional variation that we see in pomacanthity and Chaetodontity. Also, pretty clearly we have some indications that skull shape evolution has been directly correlated to functional evolution in this group. Again, something that we're going to explore as we kind of flesh out our biomechanical protocol. So a lot of us here are involved in the biodiversity community, and I would say that right now is a really exciting time for us. Our resolution of the Tree of the Life is kind of focusing more and more all the time, and our ability to quantify and compare and map the phenome of organisms is also improving. It's getting cheaper, it's getting easier. Pomacanthids and Chaetodontids are really a treasure trove of opportunities to take these really rich data layers and put them onto trees. So, if you were another person who is interested in these sorts of questions, I really hope you're going to stop by the FutureFi poster this evening so we can have a talk about what you're doing in your group and different ways that our communities can kind of work together to push this sort of data layer integration to the next level. So I guess that pretty much sums it up. Oh, and my acknowledgements went away, but I'd like to thank my PI and the rest of the FutureFi PIs for letting me come here and chat with you all today, as well as the excellent undergraduates that have contributed to a lot of this data collection in the Westgate Lab. So, thank you very much.